Hey guys, welcome to the second lecture video in our DNA chapter. This time we're going to be taking everything we learned about nucleic acids and applying that to DNA replication. Now we're going to look at prokaryotes and contrast how eukaryotic replication differs from that. Now, to start off, let's talk about the flow of genetic information. Flow of info. All right, so sequences of bases in DNA encode genetic information. Replication is the process of duplication of the DNA. So DNA replication requires RNA. It requires RNA primers. We also know that it involves polymerases, and we looked at that in the last chapter. Transcription is the process of forming RNA from a DNA template. The base sequence of DNA is reflected in the base sequence of the RNA. So the RNA will have the same genetic information that the DNA has. So DNA encodes the mRNA is in turn going to encode amino acid information. And then we have translation, which is the process of taking those mRNAs and making proteins, so protein synthesis. The amino acid sequence of the protein reflects the sequence of bases in the gene that codes for that protein. So that's translation. And we'll look at transcription and translation more in the next week. Now, retroviruses are viruses in which RNA is actually the genetic material and not DNA. And so it was thought that DNA, we talked about this in the last chapter, DNA was the starting material for everything. Turns out RNA is actually easier to make. You need to make RNA before you make DNA. And also RNA is sometimes the genetic storage information carrier. So in viruses, certain retroviruses, they have RNA as their genetic material rather than DNA. And so this reaction of taking the RNA and making DNA out of it is called reverse transcription, and it's catalyzed by an enzyme called reverse transcriptase. And then those retroviruses then take that DNA that they just made from their RNA, and then they use that to make proteins. All right. We've talked about replication a little bit of the DNA. At least, actually, that's not true at all. We're going to talk about it here. So we haven't talked about replication of DNA. We're going to talk about all that. What we did talk about in the last chapter was we looked at the nature of the nucleotides, DNA versus RNA. Okay, so one of the first things we know about replication is that it is what we call semi-conservative. Now, I'm going to talk about that in a second. So naturally occurring DNA exists in single-stranded and double-stranded forms, both of which can exist in a linear and a circular form. It makes it difficult to generalize about all cases of DNA replication. In prokaryotes, it's circular. In eukaryotes, it's linear. So we'll probably just talk about circular DNA, replication of circular double-stranded DNA, and then we'll show how those are different from eukaryotes. Now, the most details that we have were first investigated in the prokaryote, in prokaryotes, E. coli was one of the first. We know a lot about yeast also now, which helps elucidate some of the differences with eukaryotes. Now, achievement of continuous unwinding and separation of the two DNA strands is one of the challenges. And then protection of the unwound portions is also a challenge. We'll see how the body protects against those exposed base pairs. Synthesis of DNA, synthesis of the DNA template, synthesis from the DNA template is completed in the 5' to 3' direction. We learned that in the last chapter. We saw that that's the 3' hydroxyl is the free sort of tail end of our growing polymer, and then the 5' phosphate group is what comes and adds to that. So it grows from the 3', nucleotide-wise, 5' down to the 3'. The template that we're going to look at has a 5' to 3' strand, and also a 3' to 5' strand, right? The complementary strand. We're going to need to make sure we're reading the right one. One of them codes for genetic information, the other does not. Guarding against errors in replication by ensuring that the correct base pair is added to the growing nucleotide chain is one of the other challenges. So we'll talk about how what kind of protective proteins we have to help unwind and protect our DNA, and also how we can guard against errors, mismatched base pairs. So, excuse me. Semi-conservative. So DNA replication involves separation of two original strands and producing two new daughter strands. And you can kind of see that happening here. Now, we've learned a lot about how this happens by using N15 labeled nucleotides. So what we end up seeing here is that if we grow bacteria or we grow something, an organism in some medium that's rich with N15 labeled molecules, that gets incorporated into the nucleotides. And so then we have a way of sort of tracking that N15. Now you can also see that we can grow them in N14. And what happens is, if there is, wait, how does this go? The E. coli were grown in the N15 ammonium chloride as the sole nitrogen source. And so all newly formed nitrogen compounds got labeled with N15. And then they basically let them grow. It looks like they might have been mixed. Okay, so let me give you the bottom line. I don't know the experimental details, and it's hard for me to grab them all from looking at this this picture. It's not. I didn't even give you a label, so I can't really help with that. So let me just explain the two pictures here. The one on the left is showing that we have a parental strand of DNA, and that when this thing splits, both of those parental strands get complementary base pairs. So this one grows a blue strand to complement it, and this one grows a blue strand to complement it. And so then after the first round of replication, we end up with each DNA strand right now in our two cells that only has half of what they had before, half of the original strand. And then again, we can see that after another round of replication, each of those new daughter cells that came from this cell, they each only got one half of the genetic information. The G0 went to this side and the G1 went to this side. So we call it semi-conservative. The reason we know that this is the case is because if we see, if we let bacteria get N15 into their DNA, we would assume that all of the DNA that they made, let's say the double-stranded helix, should be made out of N15. So then after some time, you switch it back to N14 and you let them replicate. And what will happen is those two strands of N15 will split, and then all the two complementary strands will be made out of the N14. Later, they centrifuge this to see the different banding, and we see that if it had been solely N14 DNA, it would band near the top. If it was solely N15 DNA, it would band near the bottom. But what we see in both of the daughter cells is that it bands right in the middle, indicating that one of their strands is N15 and one of their strands is N14. Apologize for the little confusion I had trying to remember, trying to give you some of the experimental details. I don't remember the details at all, but I do remember the outcome, and that's the outcome, is that from these types of experiments, we can basically prove that this is the way it happens. DNA splits, one template gets copied to make a new daughter strand, and then that is what we call semi-conservative. All right. So let's talk about direction of replication. So DNA double helix, it unwinds at a specific point called an origin of replication. Sometimes they call them ORIs. Origin of replication. Now, polynucleotide chains are synthesized in either both or one of these directions from the origin of replication. And DNA replication is actually bidirectional in most organisms, as you can see by these back and forth arrows. At each region of replication, there are two replication forks. Now, a replication fork is the point at which new polynucleotide chains are formed. So taking a look at these two types of DNA, here you see a circular DNA with one origin, and when that replication fork starts, it actually goes in both directions, bidirectional, until they meet on the other side, and we would have now two circles of DNA. As far as linearized DNA goes, we can have multiple origins, and then as those bidirectionally fork out, you see that they will eventually meet and eventually split. And so then that's how multiple replication forks or one replication fork and the bidirectionality of replication sort of works. Now they call these theta structures. I believe this is theta. It's been a long time since I've used a lot of my Greek letters, so forgive me. But bubble or eye is what it kind of looks like. You can see that here, right? So this is a newly synthesized DNA between the regions of original DNA, and we see these theta structures. So again, in prokaryotes, one origin of replication, and eukaryotes, several. So DNA polymerase. DNA polymerase is going to be the enzyme that actually catalyzes this type of reaction, makes the DNA polymer. Now, the last nucleotide added to a growing chain has the 3' hydroxyl, okay, on the sugar group. And it's going to act as a nucleophile by attacking the phosphorus that's adjacent to the sugar on the incoming nucleotide, which has the 5' phosphate. So this causes an elimination of a pyrophosphate. That would be these guys. And the formation of a new phosphodiester bond. So DNA polymerase comes in here and catalyzes this reaction. Now, let's talk about the modes of polymerization. So there are two strands. There's the leading strand. Now this one, the leading strand, you can see here in pink, right? We're talking about the pink strand. So we've got two template strands, but we've got two polymerizing strands. So we have a leading strand. This one is synthesized continuously from 5' to 3' at the replication fork, and is exposed to the 3', 5' template strand. So DNA polymerase goes along and reads this template from 3 to 5 so that it can write, or so that it can encode, or polymerize this growing polynucleotide strand from 5 to 3'. You're going to see that a lot of the polymerases have this 5 to 3' activity. Then we have the lagging strand. So the lagging strand, you see, has to be built on an oppositely oriented strand. So the lagging strand is actually synthesized in semi-discontinuous fragments called Okazaki fragments. Now the 5' end of each of these fragments is actually closer to the replication fork than the 3' end. And these fragments end up getting linked together by an enzyme called DNA ligase. We'll talk about that towards the end. Now the issue arises from the fact that we usually have, like I said, DNA polymerase moving in the 3' to 5' direction. So it'd be sitting up here going this way. Now the problem, as you can see, is that that same polymerase needs to be reading this direction as it's synthesizing both of these strands. And so the body has come up with a really ingenious way of looping this template strand back on itself so that we can have, so if we loop it, I'm sorry about kind of flipping this around on you. If we flip it around, let's see if I can orient myself. So this was, I'm trying to figure out my direction still. If this is my 5' side. This is my 5' side and this is my 3' side. Right, because it's a big loop. So by looping it around, we notice that the 3' to 5' side is in the same direction as this 3' to 5' side. So by looping around this template strand, we can get it to face the same direction for a little stretch. And that way DNA polymerase can come in here and write a 5' to 3' Okazaki fragment. And then we have to reloop, and so that's why these are discontinuous. Okay, there's a nice video on YouTube that you can watch that'll show you this actually. One of those videos done by like Harvard or MIT or something where it's all computerized. All right, so the replication fork in both of these is moving in this direction, and so that's the problem. Both of these strands need to be synthesized here at the replication fork, but DNA polymerase needs to be moving in the same direction, and both of these strands are moving in opposite directions, and so that's the problem. Okay, let's talk a little bit about DNA polymerases. Now, enzymes that form DNA from deoxyribonucleotides on a DNA template are called DNA polymerases. So DNA, because they use the deoxyribonucleotides as opposed to ribonucleotides, which would just be RNA polymerases. And they use a DNA template. If they used an RNA template, they would be, again, different types of polymerases. In E. coli, there's at least 5 different polymerases. I've got 3 of them here in this table. 3 have been studied extensively, and so that's why we'll talk about those. DNA polymerase 1, this is a single polypeptide chain. I don't know if all of that information is on here. DNA polymerase 2 and 3, these are multi-subunit proteins. So it doesn't say anything about subunits, but this guy has 1, and these guys have multi-subunit. Now, the things that we like to consider, the things that are important as far as comparing between these are the speed of the synthetic reaction, or the turnover number. We talked a little bit about that earlier in a different lecture, turnover number, enzyme kinetics. You can see here that pol 3 has a much higher turnover number than the other 2. So it can actually incorporate, what is it, about 500,000 base pairs per minute. That's incredibly fast. So processivity is the number of nucleotides incorporated actually before the dissociation of the enzyme. So before polymerase 3 comes off of the DNA, it's put about 500,000 base pairs on. Whereas DNA polymerase 1 and 2, under 1,000 or under 2,000, and these things drop off. That doesn't mean they can't climb right back on, but they don't stay on there. They're not as long-lived in the job. Okay, now I do have a molecule here that you can look at. This is a DNA polymerase and all the different domains. So you can see here in red and yellow, green, purple, the different types of domains. With zinc fingers, I don't know if you remember, I think we talked a little bit about proteins that can bind DNA. Zinc fingers are those regions that actually like to latch onto the DNA. Polymerase active site, you can see that it's got the C terminus and the N terminus here. Thumb and palm, that's a little weird. They kind of show it as a hand, a palm, a thumb with some fingers. Okay, it's got an exonuclease site. This is where it can actually cut out base pairs. The polymerase site is where it can put in base pairs. Okay, moving on. So that's DNA polymerase, at least one of the versions from prokaryotes. Okay, so let's talk about the DNA polymerase reaction. So DNA polymerase cannot actually catalyze what we call de novo synthesis. That means it can't just start making a DNA template or a DNA polymer. It actually needs to have something to attach the first base pair to, the first deoxyribonucleotide. So it needs something called a primer. So we need primers. And there's a primer right here, so you can see that. The primer that would be on this strand would have been way at the beginning. We wouldn't see that. But on the Okazaki fragments, because every little stretch needs to be started, they all need primers. And so you can see there's one primer here, ready for DNA polymerase to start adding, this orange strand to. And then we've got this green primer up here, another one ready for, again, more polymerase activity. And then we've got a third primer right here that hasn't been added on to yet. So DNA polymerase requires an RNA primer. So a short stretch of RNA hydrogen bonded to the DNA template. You also need all 4 deoxyribonucleoside triphosphates. So lots of DTTPs and DATPs and DGTPs and so This process requires magnesium 2+. We're going to have some cofactors here. We need a DNA template. In this case, the strand of DNA provides that. And then we also need all 4 ribonucleoside triphosphates. ATP, UTP, GTP, and CTP. And this is for various energy purposes. And then also I think the ribonucleoside triphosphates may be the source for the deoxyribonucleoside triphosphates. Actually, I'm not sure what the RNAs are needed for. Maybe just for the primers. Okay, we're going to have a few different DNA polymerases in action here. Let me see. No, we don't need to look at that yet. Okay, so DNA polymerase 1, it has a job that kind of focuses on repairing and patching. So it's a repair enzyme. It can go and actually see if there's any nicks. It can see if there's any mismatches, and correct those. And we'll talk about how that happens in a little bit. We have DNA polymerase 3. Pol 3 is our main. We saw this had the highest processivity, highest turnover. This is the one that polymerizes the newly formed DNA strands. And then DNA polymerase 2, 4, and 5 tend to act as repairing enzymes of some sort. And they have a few exonuclease activities that are part of the proofreading and repair functions. Proofreading being removing the incorrect nucleotide, and then repair would be replacing them with the correct ones. Now DNA replication can also be carried out by replisomes. Now I think I have a picture. I don't know if I have a picture of a replisome. I think this is the replisome. This whole bit right here. So let's talk about that. So a replisome is the complex of the DNA polymerase, the RNA primer, primase, okay, helicase at the replication fork. So we need a few of these different enzymes. We'll talk about what these all do, and sort of I'll just kind of summarize it all for you guys. We have DNA gyrase. You can see gyrase in here. I think primase is the enzyme that comes in here and actually helps to unwind the DNA. So we've got some type of keeping the strands apart, putting on the primer. So primase is putting on these RNA primers. Helicase is helping to unwind. We have DNA gyrase. DNA gyrase is a class 2 topoisomerase, if you remember, that was involved in adding or removing supercoils or creating supercoils ahead of the replication fork to help unwind and relieve some of that pressure or that torsion, that strain. And the energy required for these processes are supplied by the hydrolysis of ATP. So that's why we needed to have some ATP in there as well. So again, DNA gyrase, helicase, and primase are involved right here at the beginning of the replication fork to help open up this whole process. Okay, so we talked a little bit about how prokaryotic DNA was negatively supercoiled, and so opening the helix requires introducing positive supercoils ahead of the curve. Now, DNA gyrase also fights against positive supercoils by placing negative supercoils ahead of the replication fork. So we've got a lot of concern and care taking to make sure that the DNA unwinds properly. Okay. Now, DNA gyrase also has a role in creating this little swivel point at the neck that opens and reseals. It opens and reseals this over and over, ensuring that the newly synthesized DNA will automatically assume the supercoiled shape. Okay, helicase. Helicase, I mentioned, is a helix destabilizing protein, and it unwinds the binding at the replication fork. We have SSBs. These are single-strand binding proteins, and these stabilize the single-stranded region by binding very tightly to them. So they prevent any other unwanted side reactions happening to our exposed nucleotides. And then again, primase is the enzyme that makes the short section of RNA primer. So we have a primosome. Now, a primosome is the complex at the replication fork that consists of the RNA primer, the primase, and the helicase. So a primosome is just this little bit right in here. The replosome obviously needs more. Replosome has DNA polymerase, it has the template, and then it also has a few of those other proteins. Okay, DNA polymerase 3 commences the synthesis. Newly formed DNA linked together 3' to 5', starting with the primer. And then it goes on that way for the leading and lagging strands. Now, as the replication fork moves away, the RNA primer is removed by DNA polymerase 1. So all of these primers down here, right, on the lagging strand get removed by DNA polymerase 1, and then replaced with regular deoxynucleotides, and again by DNA polymerase 1. And then DNA ligase, so ligase here shown in yellow, this is responsible for actually linking together all of the different groups, because what DNA polymerase can do is it can come in here and it can actually add, let's see, it can actually add, what I'm trying to say here is that this image isn't great. This orange strand here growing off the primer, it'll get added all the way up until this green bit right here, but these two can't be joined together by DNA polymerase 1. DNA ligase has to go and, well, first DNA polymerase 1 will remove the green primer, but there will still be a gap right here, a gap, and ligase seals that. Okay. Okay, so that's basically it. So that's the parts that we want to talk about. DNA polymerase's job, polymerase 1 job, ligase's job, primer, helicase, SSBs. We can talk a little bit about this cool beta subunit sliding clamp business, because this is pretty neat. So let's see, I think I have a picture of that here. So what we've got here is actually the clamp loader. Now, actually, before I talk about the clamp loader, one more thing I want to say about this. We recently, last 10 years, have found out that there's actually a few DNA polymerase 3s working here, and you can see them here. So we actually have 2 DNA polymerase 3s working here on the kind of like as a group. One of them's on the leading strand, 2 of them are on the lagging strand. So these 2 are kind of connected together, but all 3 of them are in this replosome. One DNA polymerase 3 is dedicated to the leading strand, 2 to the lagging strand. So not that there's just one DNA polymerase 3, there's actually 3 of them working. All right, this is the clamp loader. This is part of the polymerase 3 enzyme that opens, and then there's a thing called a sliding clamp. So the clamp loader opens the sliding clamp and then allows the DNA, excuse me, the DNA to actually be fed through these little things that keep them open. If you notice, it was a double-stranded going into a single-stranded area here. Helps keep the DNA unwound and separated. This is a pentameric enzyme. There's 5 parts. That's a member of the ATPases called the AAA plus superfamily. Anyways, this is in prokaryotes, and then these 2 pictures are the equivalent in the eukaryotic DNA. So you see, it's not a pentamer over here; it's a tetramer. 1, 2, 3. But it does the same sort of job. It is a sliding clamp, so they say. Thought I'd show that. It's pretty neat. We'll talk a little bit about the eukaryotic one when we get to just a little bit of those differences. Okay, proofreading and repair. So DNA replication takes place only once each generation in each cell. Now mutations, we talked about a little bit how those happen, the frequency or so around the chromosomes, beads on a string, remember? Mutations are errors in replication that occur spontaneously, about once every 10 to the 9, 10 to the 10 base pairs. So every billion or 10 billion base pairs. And can be lethal to the organism depending on where that mutation happens. Now, proofreading is the removal of the incorrect nucleotides immediately after they're added to the growing DNA. And then the errors in the hydrogen bonding is essentially how the DNA knows this. We'll look at a couple. And errors in hydrogen bonding lead to errors in the growing DNA chain about once every 10 to the 4 to 10 to the 5 base pairs. So what we've got here is one of the proofreading pictures here. So you can see DNA polymerase 1 is moving along the template strand, and it's reading the pairs. Now what we see here is that there's a GT base pair that's mismatched. And so what DNA polymerase 1 has is 3' to 5' exonuclease activity. That means it can actually go backwards direction, right? DNA polymerases are usually, or at least polymerase 3, we know that it catalyzes the 5' to 3' direction. But it can also, this one, 3' to 5' exonuclease, meaning it can go back and cut out this one that it just put in. So it can catalyze it in, and it can also cut it out. So that would be an example of that exonuclease activity. Now, we have two different types. Actually, I think we only have one type. It's called Nick translation that we're going to talk about. Now, this is a cut and patch process catalyzed by polymerase 1. It takes place during replication. It involves cutting so removing the RNA primer by the 5' exonuclease activity, and then patching is the incorporation of the required DNA into the chain by the same enzyme. So if the same enzyme is cutting and patching, then it's called Nick translation, and it's essentially the removal of it's the removal of an RNA primer or a DNA mistake by polymerase 1 using 5' to 3' exonuclease activity. Nick translation. So let's take a look at it. So in this previous one here, you can see that there's a mismatched base pair. So let's move on. So let's say that this one also has a mismatched base pair. You can see it here, T to C. So what DNA polymerase 1 will do is it'll move along here, detect the mismatch. It'll back up, cut it out, and it can actually cut out a long stretch. It can remove up to 10 nucleotides in the 5' direction downstream from the 3' nick. And then if that same enzyme goes and then fills the gap, it's called Nick translation. What we see here, because remember DNA polymerase 1 can't actually link anything together, once it cuts that out and then it removes a bunch of base pairs and then fills that gap, the nick actually got moved. So here was the original nick, now the nick's down here. So translation in that it got shifted over. Now DNA ligase will still have to come in here and actually patch that up. Okay, so that's just one of the ways that this happens. There are lots of exonuclease type of activities, but DNA polymerase 1 is one of the main examples of that. Let's talk about mutagens. Mutagens are agents that bring about mutations. They include ultraviolet light, which can create pyrimidine dimers. So these are two thymines near each other in the DNA backbone. You can see UV light can cause covalent bonds to form between the two of these. So pi electrons from the two carbons on each of the two pyrimidines, they form a cyclobutyl ring, and that distorts the DNA's shape, prevents replication. Ionizing radiation can do this, anything that's enough to knock electrons off. There's also various chemical agents or free radicals that can actually cause damage to DNA. Here you can see a free radical coming up on a base pair. This is the base backbone. Here's the base DNA backbone, and this free radical can actually come in here and hydrolyze all of this. It can actually break up that sugar bond, breaking it apart, freeing the base. So that's bad news. Destroying the sugar rings. All right, so let's talk about some of the other repair mechanisms. So the first of the repair mechanisms is called mismatch. So I was showing you Nick translation, which is part of the DNA polymerase proofreading. Now, mutations can also happen, and damage to DNA can happen after the fact. And so when that happens, we need to be able to go and repair. So repair is slightly different from proofreading. So as far as repairing goes, we have mismatch repair. Now mismatch repair, there's an enzyme that'll recognize that two base pairs are incorrectly paired. So the first step that happens is that we gotta spot an incorrect pairing. the area of the mismatch gets removed, and the area gets replicated again by DNA polymerase. Now the challenge is, which of the two strands is the correct one, right? Which of the two strands is the one that you're supposed to be using? My question is there supposed to be a T here and this should have been an A? Or is there supposed to be a G here and this should have been a C? That's the question. And there's actually a neat way. There's a small window of opportunity that comes up immediately after replication to be able to spot a mismatch. And what happens is the longer DNA exists in your body, it gets methylated. So right here, you can see one of these strands actually has a methyl group on it. The methyl group indicates that this is the template. So the first thing that happens is there's a couple of enzymes, mute H, mute S, mute L. They find this, they link the mismatch with the nearest methylation site. So this gets folded over. So that's what's happening here. This is folding this way. A protein is associating with this methylation site. Actually, it gets folded this way. My bad. Folded that way, so that the blues are on the inside and the reds are on the outside. This G gets brought near this methylation site. So now they're linked, and then the reds get cut off, because that's the mismatch. So the reds get cut off, and then they get re-synthesized. So they re-synthesize all of this. And then, again, as time goes on, those will both get methylated, and then during the next round of replication, that's how the body will know where the right, or what the correct template is. So mismatch repair. Let's talk about base excision repair. So, and that's this guy over here. Damaged base can first be removed by DNA like glycosylase, and this leaves what we call an AP site. It's apurinic or apyrimidinic. Yeah. So basically it's missing a base. So the base gets removed and now it's missing. This was again done by glycosylase. Then the sugar and phosphate backbone are removed by an AP endonuclease. So we go in here and we actually cut out the backbone. Several more bases are removed by excision endonucleases, or exonucleases, sorry. And then DNA polymerase 1 will come in and fill the gap, and ligase will seal the phosphodiester bond. So damaged base identified, removed, backbone removed, more room made, gap sealed. Base excision repair. Nucleotide excision repair. So bases were removed first, nucleotide, the whole thing's going to come out. So this is common for DNA lesions that are actually caused by the UV or the chemical means. So the pyrimidine dimers are a perfect example. The section containing the lesion, it gets removed by an ABC exonuclease. And then, so you can see, gone. And then DNA polymerase 1 and ligase come in and fill the gaps. And then we have something called double-stranded breaks. So this was nucleotide excision. This is double-stranded breaks. This is when breakages of both strands of DNA happen. This is a real big threat to the stability of the genome. Because how do you know where to reattach it, right? And what's missing? Was there a big chunk missing? I mean, who knows? This is bad. The repair mechanisms actually include a few different processes. One called recombination. We'll talk about that. The other called non-homologous DNA end joining. Non-homologous DNA DNA end joining, NHEJ. Yeah, and so these bits of DNA are just linked back together. All right, we can talk a little bit about genetic recombination. So this is a natural process in which genetic information gets rearranged to form new associations. There's homologous recombination. This involves mixing of homologous sequences, so two genes that code for the same thing, for example, or non-homologous recombination. And this involves the combination of different nucleotide sequences. This occurs in specific zones on the chromosome called hotspots. We'll look at that in a second. First thing I wanted to show you was actually just a physical exchange of chromosome parts that happens during viral recombination. So here, two viruses infect the same cell. We've got a heavy phage and a light phage, just to kind of mark them as different. Their DNA goes inside. Recombination happens when the packaging of these virus particles happens, and so we get some DNA that's just got heavy phage, some DNA that's actually got some mixed heavy and light phage, and some light phage DNA. So the recombinants are the ones that have the mixed DNA. Now this was just physically packing them together. There wasn't any kind of crossing over or some weird business happening. Just the sorting. So the recombination via sorting. Let's look at the holiday model. Now, holiday, I think your book goes into depth on this. Now, the holiday model describes how recombination occurs by the breakage and reunion of DNA strands, so that physical exchange of DNA parts takes place. So this involves a few steps. The alignment of homologous DNA segments, the nicking of the same place on the two strands, so nicking and nicking. Strand invasion, we call it. DNA of the two strands kind of swap places or cross over. So I mentioned crossing over. This takes place during gamete formation. So when your body's getting ready to divide and to make your haploid cells, your eggs and your sperm cells, there is some amount of recombination that happens where new genetic variability is introduced. In addition to, there's also some sorting that goes on, right? Your chromosomes from your parents got kind of randomly sorted and then separated so that you got some from your dad, some from your mom, but there was also some parts of those chromosomes that crossed over, making new sequences that were different from both your parents. And so that's this process. So after the strand invasion happens with some crossing over, we get ligation, so sealing of those bonds. Branch migration, this little crossover kind of moves to a position down the chromosome so that this can now bend and give us this weird little exchange cross area. And so then you can see here, there's a few cleavages that happen. Now, what we're seeing here are the two different ways that this can happen. This can just get cut right here, and we would end up with two strands. We'd end up with this top half and this bottom half as depicted here. So the plus strand and the plus strand of these two didn't actually get mixed up, but the minus strands got mixed up. There's another version where this also gets folded over, and we end up with the plus strands also getting swapped a little bit too. So this, again, just two different ways of ending this process, but different regions of DNA get mixed with each other. Now, eukaryotic replication. Let's talk about that. Now, this is just like prokaryotes, but it's more complicated because of a few things. It's more complicated because of the multiple origins of replication. It's more complicated because of the need to control the timing to that of the cell divisions. And it's also more complicated because it involves more proteins and more enzymes. Cell growth and division is divided into a few phases. We've got M phase for mitosis. We've got this G1 phase of rapid growth. We've got S phase. This is where the DNA replication happens. And then G2 phase, growth again and preparing for cell division. So we're going to kind of couple that to the different parts of our replication here. So eukaryotic replication can only be initiated by chromosomes from cells that have reached the G1 phase. So once we've got to G1, as you can see here, we are ready to possibly start replication. Step 1 involves various proteins. It involves the origin replication complex, or this guy. This is bound to the DNA throughout the cell cycle, but it serves as an attachment site for the several proteins that are going to help control replication. We need the replication activator protein, RAP. This protein here is going to, when it binds, it'll help prepare again the DNA for the start of replication. And then we need replication licensing factors, copyright infringement and all that stuff. Just kidding. Replication licensing factors here are proteins that are essential for DNA replication. Some of these are cytosolic, meaning that they float around in the cytosol. Others are found in the nucleus. So all of these guys together form the step one of this, the pre-replication complex. So the pre-replication complex, DNA, ORC, RAP, RLFs. Step two, this involves some other proteins and protein kinases, and then some proteins called cyclins. Now, cyclins are proteins that are produced in one part of the cell cycle and degraded in another. This is good because that means that they can actually, the activation of these cyclins serves to initiate DNA replication, and then when they get broke down, they stop another pre-recognition complex from forming. These cyclins are combined with cyclin-dependent protein kinases, CDKs, cyclin-dependent protein kinases. So these cyclins will get phosphorylated and dephosphorylated as needed to help start this process, but also they get broken down so that they don't keep doing this process. So they kind of show up right around here in the phase, or in this phase. Okay. In the G2 phase, DNA has been replicated, and so we would already then have our DNA daughter strands. And then the DNA gets separated during mitosis into daughter cells. And so this actually, there's a process that happens. The nuclear membrane actually dissolves, and this allows the entrance of the RLFs that are produced in the cytosol. This makes sure that each new daughter cell can actually have RLFs present in their cells and in their nucleuses for new rounds of replication when they mature. Okay. Let's take a look at some DNA polymerases for eukaryotes, just to see that there are some different ones. So there's at least 19 different polymerases in eukaryotes. 5 have been studied most extensively. Alpha, beta, it looks like gamma, delta, epsilon. You can see here a few different things. Polymerase A makes primers. So this is the guy that makes the primers. Polymerase delta, this one, this is the principal DNA polymerase. So this is like POL3. In that it's the main one. We don't have turnover here or processivity to look at, but that's okay. Polymerase epsilon, this one is involved in the leading strand replication. It may replace polymerase delta in the lagging strand synthesis. So these two have similar functions. This is a repair enzyme, beta. And polymerase gamma, this one carries out DNA replication in the mitochondria. Yeah, okay, so that's what these guys do. As far as the exonuclease activity goes, you can see that too. So 3 5' exonuclease. So this is like what POL1 could do. And you can see here that POL3 and epsilon and this guy can do that. But it doesn't look like... Oh, my bad. 3', yeah, 3' to 5'. That's backwards. Yeah, that's backwards. It's odd that this guy doesn't have that activity, and he's a repair enzyme. So I wonder what kind of repair that one does. Well, obviously it does 5 to 3' repair. Actually, maybe we can't associate exonuclease activity completely with repair, because it looks like, you know, this guy does repair, but it doesn't have exonuclease activity, so it must repair through another mechanism. But it's at least worth noting that all of these, right, can do 5' to 3' polymerization activity. Some of them can actually go backwards and cut out. Okay. And I mentioned all this stuff here. Oh, these are some comparisons. So this is a nice comparison between prokaryotes and eukaryotes. So we can see some of the similar functions. The polymerases in the prokaryotes are also exonucleases, but not all the polymerases are exonucleases with eukaryotes. Okazaki fragments are about 1,000 to 2,000 residues versus 150 to 200. No proteins. We have histones. Okay. And again, This is the basics of the eukaryotic replication fork. So again, not many differences here, other than that we've got topoisomerase instead of helicase, or instead of DNA gyrase. We've got we still have helicases. We still need primases and primers. We still need single-stranded binding proteins. They're called RPAs here. We still need a leading strand. We still have a lagging strand. You can see that there's some other bits in here. Now let's just talk about telomerases while we're at it. So replication of linear DNA poses a particular problem at the ends, right? At the ends of the molecules. They don't have, they're not circular. Telomeres are the structures then that are going to be at the ends of linear DNA. And we only find them, again, in like eukaryotic organisms. Now, the telomeres on the ends are a series of repeating DNA sequences. They're non-coding. They basically act as a buffer against the degradation of the DNA. Telomerase, yeah, I'm saying that right. Telomerase is a ribonuclear protein that contains a section of RNA that's actually complement to those repeating structures that we find in the telomere. So the telomerase has RNA in it that recognizes those long strands at the end of the DNA. This provides a mechanism for synthesis of telomeres. And what's really interesting is that these get reactivated somehow in cancer cells. Cancer cells seem to be very good at keeping their telomeres long. There is some kind of information that maybe couples the shortening of telomeres to basically getting old and dying. If we can keep our telomeres long, there might be some chance of living longer or something like that. Actually, that is the end of the lecture here. So, all right. So again, focus on the big picture stuff. Don't get caught up in the details between prokaryotes and eukaryotes. They are very similar. A lot of the same general processes are happening. All right, guys. That does it for this week. Next week, we're going to look at protein synthesis, metabolism, and photosynthesis. So good luck on exam 2. See you next time.